So next up, we're going to talk about autosomal recessive patterns of inheritance and disorders. So we're going to talk specifically about genetic disorders. A lot of times they're going to be caused by recessive alleles. In the event that we are talking about a recessive disorder, an individual who is homozygous dominant will not have the disorder. An individual who is heterozygous will not have this disorder, but is often going to be a carrier for the disorder. And an individual who is homozygous recessive is going to show that disorder. So, for example, if both parents carry one copy of the recessive gene, they are unaffected, but they are capable of having a child with two copies of the gene who will be affected. So a carrier is someone who displays the dominant phenotype but is heterozygous for a trait. They're going to carry the recessive allele and can pass it on to descendants. Cystic fibrosis is one of the most common recessive autosomal disorders that is also lethal. About 1 in 20 individuals are carriers, but only about 1 in 2,000 actually have the disorder because it is so rare that a homozygous recessive individual will present. Cystic fibrosis is caused by a defective chloride channel carrier protein that is encoded by the gene GFTR on chromosome 7. Research has shown that chloride ions simply cannot pass through those defective channels onto the plasma membrane. And the movement of chloride ions is very important because it creates an unequal concentration gradient that forces water and sodium ions to follow behind it. The lack of water can cause an abnormally thick and sticky mucus to build up on the lung epithelial cells, preventing them from functioning normally. There have been many recent advancements in gene therapies for the treatment of cystic fibrosis that have resulted in increased life expectancy for individuals with CF to about 35 years, but it is still ultimately a fatal disorder. So that's just a little bit background on cystic fibrosis and how that can be related to genetic disorders. Sickle cell anemia is another genetic disorder that is an autosomal recessive genetic disorder, and we're going to use that as an example in some of our labs that we do later on. So the dominant allele is often produces, in the case that we're talking about a recessive disorder, the dominant allele often produces a functional protein that the recessive allele does not. Another example of this would be albinism. Albinism is lacking the ability to produce brown pigment melanin. The ability to produce melanin depends on the enzyme tyrosinase. Tyrosinase is formed by a dominant allele that results in the pigmentation produces functional tyrosinase. Recessive allele, it results in albinism, produces the non-functional tyrosinase. So without that correctly formed enzyme, we cannot produce the melanin necessary. We're going to look at some of the tools that can help us sort of track genetic traits would be pedigrees. A pedigree is a chart that shows genetic connections among individuals in a family. This is especially useful in following recessive alleles that are not visible in the heterozygote. So even if we, lose sight of them physically, we can still sort of track them. So here is a pedigree showing the inheritance of a dominant autosomal trait and a recessive autosomal trait. So in this case, we're going to see that this particular genetic trait is dominant. So in the presence of a heterozygote, we can actually see it being passed down. So we know that this individual right here is going to be heterozygous if we're going to cross a homozygous recessive with a heterozygous individual. We'll see that these individuals over here are all recessive for that particular trait. There are going to be some exceptions to what we have found to Mendel's work. So although Mendel studied relatively simple systems, his principles can be applied to complex patterns of inheritance, such as incomplete dominance, codominance, multiple allelic traits, pleiotropy, or polygenic inheritance. In the classic case of complete dominance, any heterozygous individual will exhibit traits associated with the dominant allele, but not those of the recessive allele. So complete dominance. Heterozygote exhibits the trait associated with the dominant, but not that of the recessive. However, this isn't always going to be the case. What we found in the case of codominance, effects of both alleles are apparent in the heterozygote. Great example of this would be blood type AB. blood type A is dominant, blood type B is also dominant. Together, when you have both A and B blood types, you're going to see both A and B antigens. The protein products of both the A and B alleles are going to be expressed on the surface of the red blood cell. Incomplete dominance is when the expression of a trait in a heterozygous individual is in between the way the trait is expressed in a homozygous dominant or homozygous recessive person. So a great example of this would be a cross of a red flower and a white flower and then producing a pink flower as a result. Or another example of this is going to be sickle cell allele. The heterozygote has the sickle cell trait, but has sickle cell trait, but is not necessarily sick with sickle cell anemia. They're a carrier. So there are, in this case, the phenotype is going to reveal genotype. A great example of this, like I said, would when we cross a red flower with a white flower. We find that when you have the red flower and the white flower allele, you end up with a pink flower. Pleiotropy. Pleiotropy is when one gene has many different effects. Besides providing an example of incomplete dominance, sickle cell anemia is an example of pleiotropy. The sickling of red blood cells is caused by abnormal hemoglobin that affects a lot of areas in the body. So, sickle cell anemia trait or sickle cell trait doesn't affect just the shape of your blood cells, but can actually affect many other parts of the body. Those sickle cells can break down, clog vesicles, accumulate in the spleen, and wreak havoc in many areas of the body. These effects can also affect the heart, brain, lungs, kidneys, muscles, and joints. So just because it's a trait for a particular blood cell doesn't mean it may also have effects elsewhere in the body. Here's an example of how that sickled cell can cause issues in all of these different systems. Another great example of this would be Marfan syndrome. Marfan syndrome is pleiotropic and it results in the following phenotypes: disproportionately long arms, legs, hands, and feet, a weakened aorta, poor eyesight. Now, it may seem a little bit weird. What does poor eyesight have to do with a weakened aorta? How is that in any way related to disproportionately long legs or long limbs? And it's because the Marfan syndrome affects a connective tissue disorder. So because it affects connective tissues, it affects the skeleton, it affects your heart and blood vessels, it can affect your eyes, it can affect your lungs, it can affect your skin. So even though these symptoms seem really weird and disparate and like they have nothing in common, they're actually very closely related because it all comes back down to that connective tissue disorder. There are also things known as multiple allelic traits. Multiple allelic traits are an example of a non-Mendelian trait. This happens when a trait is coordinated by more than one alleles. So this is when 3 or more forms of a given gene exist across many people in a population. Fantastic example of this would be ABO blood types. Blood type is going to be determined by the presence of certain polysaccharides on the surface of the red blood cells. Type A blood has the A polysaccharide. Type B blood has the B polysaccharide. Both A and B, type AB, has both A and B polysaccharides. Type O has neither. So you're not going to necessarily be, in the case of tall versus short plant, we're not looking at tall and short. We're looking at A versus B versus AB versus O versus AO versus BO. So there's many, many more possible allelic combinations going on. Here is a table that's going to help break down all the possible genotypes and what that's going to look like as a blood type. This table will be very useful to you when you are doing your Mendelian genetics worksheets. Please let me know if you have any questions, but I will be happy to run another tutoring session when we start using Punnett squares to work out these genetic problems. Polygenic inheritance. This occurs when a trait is governed by 2 or more alleles. So examples include things like height, skin color, or the prevalence of diabetes, meaning that there are certain trait, there are certain characteristics that are encoded for on multiple genes. So typically involves 2 or more genes, often on different chromosomes. Many traits, height, skin color, eye color, vary almost continually from one extreme to the next. And they can also be controlled by the environment. So this is why if you have a tall father and a short mother, you don't get children who are all tall, or you don't get children who are all short. It's because, here, we'll look at this table next. Human height occurs on a spectrum because there are actually multiple genes that encode for height. So here we have 6 different genes that code for height, and we have 2 individuals of absolutely medium height, dead across the board, because they each have 3 tall genes and 3 short genes. The problem is when they cross, it is possible, there is a 1 in 64 chance that they are going to have an incredibly tall child. That child is going to be very tall because he's going to inherit 3 tall genes from mom and 3 tall genes from dad and be super tall. It's also possible there's a 6 in 64 chance that he's going to be pretty tall. He's going to inherit 5 tall genes from mom and 2 tall genes from dad. Or, I'm sorry, 3 tall genes from mom and 2 tall genes from dad. Or 3 tall genes from dad and 2 tall genes from mom. And then there's a good possibility that the child is going to be a little bit taller than his parents, a little bit taller. We're going to see 3 tall genes from mom and 1 tall gene from dad, or vice versa. Now, the odds are very likely that this child is going to be exactly as tall as their parents. Very likely. Now, it is possible that this child could also be short. There's a 1 in 64 chance that this child will inherit 3 short genes from mom, 3 short genes from dad, and be crazy itty bitty bitty. This is because there are multiple genes that encode for height. There are also multiple genes that encode for skin color. There are multiple genes that encode for eye color. This is why it's possible to have spectrums of these various traits. There are genes that are going to be located on the same chromosome. This means they're usually going to be inherited together, and this is going to kind of fly in the face of independent assortment. Mendel developed his principles while studying genes. He looked at 7 different traits and discovered that they all assorted themselves independently, meaning that a pea that was dominant for height was just as likely to be dominant for color as a plant that was recessive for height. But this is because each of the traits he studied were coded for on completely separate chromosomes. There are some genes that are located on the same chromosome. Now, they might not be in the same location. One might be at the northern tip, one at the southern tip, or they might be right smack next to each other. The closer they are together, the greater likelihood that they're going to be inherited together. These traits are usually described as being linked. Linked traits do not usually sort independently, but they can. Why is it that they can? Usually is emphasized here because there is a mechanism that can unlink genes on the same chromosome. That is going to be those crossing over events. Crossing over events can lead to those genes being sorted separately. Sex-linked genes. The Y chromosome is going to be much smaller than the X chromosome, and it's going to carry fewer genes. Most genes on the X chromosome have no corresponding alleles on the Y chromosome, meaning that these can only be inherited from one X chromosome to the next. So these are X-linked genes. Now, they're going to actually have a different pattern of inheritance. There's going to be a recessive phenotype of X-linked genes, more common in males, because the son can inherit the X-linked recessive trait only from the mother. And there are going to be a couple of examples of this disorder. Red-green color blindness is a great example of this disorder. And we're actually going to, I'm going to give you this diagram here so you can spend a little bit of time looking at figure 20.14 in your text, but we're actually going to hold off on this discussion until we can do this in person. So stick around or tune in for our discussion on X-linked traits. The last thing I want to mention are sex-influenced genes. These are going to be autosomal genes whose expression is influenced by the sex hormone. Their expression is going to differ in males versus females, and an example of this would be male pattern baldness. This is going to be much more common in men than women because its expression depends on both the presence of the allele for baldness and the presence of testosterone. Men can be heterozygous for the trait and still show the pattern of baldness because they have the testosterone. Women who are homozygous for the trait will eventually develop pattern baldness in their life, but it'll be much later on when their estrogen levels drop and their testosterone levels increase. All right, that's it for our principles of inheritance.